Today we have for the ISYEB seminars a very special guest, Dr. Marta Munoz from Yale University. Dr. Munoz got her PhD at Harvard University in 2014. And as I just said, she's now a professor at Yale University, working on one of the most interesting questions, I think, in biology, which is why some traits and lineages evolve very fast, very rapidly, while others seem to be very slow to change. And today she's going to talk to us specifically about behavior and how that can act as a motor and a brake for evolution. So I'm personally very excited to watch that, and I hope you all are too. So without any further ado, Dr. Munoz, please.
Thank you so much, Bernardo. Before getting started, when I share my presentation, my little image is going to pop off. So before I disappear for the next 45 minutes or so, I just wanted to say thank you so much for being here digitally. And also big thank you to Anthony for inviting me to come give this talk and the opportunity to share my research. This has been such a delightful visit already, and I hope to get the chance to meet many of you later today. Okay, with that, I'm going to start sharing my presentation. Be 1 second. 1 moment. It seems to have closed. Is it Bernardo? Would you mind unmuting for a moment and letting me know if you see this?
Yeah, I don't see it yet. So recording has started, but I don't see the screen yet.
Okay, sure. 1 moment. It should be okay now. Okay, let's try again. There it is. Okay. And Bernardo, once more, are you able to see it now?
Yes, I can see it.
Okay, wonderful. Sorry about that. Okay, so as Bernardo intimated, my research program is motivated at the broadest level by a general observation in biology, which is that evolution proceeds unevenly across the phenotype. And by this, I mean that some traits appear to evolve very rapidly, perhaps even in overdrive, whereas others seem to be inert or nearly so for millions of years. To give an example, when compared to other aspects of their morphology, lizard genitalia evolve so fast that they may be deemed to be evolving at, and this is plucked straight from an actual press release, at super speed. Now, equally impressive is that some aspects of the fish mandible can evolve up to 30 times faster than others. Now, keep in mind that these are all integrated structures that need to work together for emergent functions, and yet the rates of evolution can be so different among different aspects of the phenotype. So what I'm really saying is that sometimes trait evolution gets to drive along the Autobahn, going at warp speed, whereas other times evolution is much more like driving in New York City at rush hour. But why is this? Why do some traits get to drive in evolution's fast lane, and why are others stuck in evolution's slow lane? Well, I approach this question by studying one of evolution's key architects, which is behavior. Through their behavior, organisms can sculpt their evolutionary trajectories. For example, on the one hand, organisms can shield themselves from natural selection through their behavior, and in so doing, slow the rate of evolution. By contrast, organisms can also expose themselves to greater amounts of natural selection, and in so doing, accelerate evolution. I try to study this phenomenon by dissecting the myriad ways that organisms interact with their environments and piecing together how that shapes evolutionary patterns of diversity. To explain what I mean, consider this Enolus lizard perched on a tree somewhere in the Caribbean. In order to escape predators, find mates, or acquire food, this organism interacts with its phenotypic dimensions and behavior with its environment. For example, its ability to escape predators is limited by the thermal dependence of its performance, its hydric and metabolic economy, and its morphological dimensions. And in turn, all of these features interact in complex ways. So my research centers on demystifying all of the ways that organisms interact with their physical environments and determining how that shapes evolutionary patterns of diversity. And I focus on several ecological features, but perhaps most salient among these is temperature. And I focus on temperature because in ectotherms like the Enolus lizards I'll be discussing, it's extremely important for all of their emergent functions. The ability to perform a task, for example, sprinting, is contingent on body temperature such that it's optimized over a relatively narrow range of temperatures and then decreases at higher and lower temperatures until the animal is effectively immobilized. This curve has a lot of really important features. For example, there are critical thermal limits, the critical thermal minimum and the critical thermal maximum, temperatures above and below which locomotion quite literally ceases. This temperature has a maximum performance. It also has a temperature at which performance is optimized. So my approach is to dissect this curve into its component features and determine the factors that shape their evolution. And I do so in lots of reptiles and amphibians, but today I'll be focusing on perhaps my favorite lineage of reptiles, Enolus lizards. This is a very diverse radiation of lizards distributed throughout the tropical and subtropical regions of the Western Hemisphere. There are more than 400 species total in this radiation, but they're especially diverse in the Caribbean, where more than 150 species can be found. Now, you may be familiar with this lineage because it's a classic example of adaptive radiation. On each of the major islands of the Caribbean Greater Antilles, this being Cuba, Jamaica, Hispaniola, and Puerto Rico, these convergent assemblages of microhabitat specialists termed ecomorphs have independently arisen. And I've shown here some of the representatives from different islands in the Caribbean just to give you an example of how pronounced this convergence can be. And of course, these anoles are so colorful and charismatic to work with. They have these extensible throat fans termed dewlaps, which they use in social communication and territorial defense. And they're just these jewel-toned, really fun organisms to interact with. But today, I'm going to be focusing on my preferred type of Enolus lizard, the LBL, the little brown lizard. And in particular, I'll be focusing in on the cybatoid anoles, a radiation of trunk-ground lizards. This means lizards that perch within the bottom 1.5 meters of the ground. This lineage is comprised of Enolus cybotes, shown here, and its close relatives, which are found on the Caribbean island of Hispaniola. And I promise you that what this lineage lacks in orange and blues and greens, it completely makes up for with physiological badassery. So today, I'm going to start by focusing in on this cybatoid lineage of Enolus lizards from Hispaniola to pick apart the role of thermal regulation as a pacemaker for trait evolution. I'm going to talk a lot about the role of thermal regulation in this lineage, and we're going to see some interesting patterns in this one radiation. I'm going to then zoom out to the entire Enolus adaptive radiation to ask whether the same patterns we observe in the cybatoids can characterize Enolus lizards more generally, and what this can tell us about the adaptive radiation in general. And hopefully at the end come together to discuss some classic ideas about the role of behavior and evolution with some new perspectives and see where we can go from there. Okay, so I decided to work on Hispaniola for a couple of key reasons, and one of them is that it's incredibly topographically complex. There's a lot of elevational variation on the island. In fact, you can be 3,000 meters above sea level and still be in the Caribbean, but only if you do so on Hispaniola. The island itself is dissected into two major mountain chains, the Cordillera Central in the east and the Sierra de Bauruco in the west. Now, near sea level, it is as lush and balmy and tropical as you might expect of the Caribbean. It's pleasant for both lizards and Homo sapiens. But as you go up in elevation on Hispaniola, and I mean really up in elevation, the habitat cedes to this relative moonscape of monodominant pine forests surrounded by bunchgrass fields and rocky outcrops. And it's quite cold up there. At the highest elevations of Hispaniola, it can be more than 20 degrees Celsius cooler than near sea level, which is a lot of thermal variation for humans and especially a lot to consider when thinking about small tropical ectotherms like anoles. It's not uncommon to get frost up there, even in the summer, and it's quite chilly, which I quickly discovered during my first expedition to Hispaniola, for which I was completely underprepared. But I had excellent company on the summits of these mountains in the form of Enolus lizards. On the summits of the eastern mountains, you can observe Enolus stribi, shown here on the right, and in the summits of the western mountains, you can observe Enolus armori, shown here on the left. And these two species together are part of a broader radiation, that cybatoid radiation of trunk-ground anoles from Hispaniola. Some species in this radiation are found near sea level, like Enolus whitemani and Enolus longitibialis. A few species like Stromai and Marcanoi creep into mid-elevation, get up to about 900, 1,000 meters perhaps. Now, one species, Enolus Lizards, is truly remarkable. It's essentially found island-wide, except at the highest elevations of Hispaniola, where it becomes replaced by Enolus armori in the western mountains and Enolus shrievi in the eastern mountains. I was really intrigued by this incredibly diverse radiation distributed in such different thermal habitats. So my first forays into this system were purely curiosity-based. I wanted to know how, if at all, were these tropical lizards behaviorally or physiologically compensating for such different thermal environments. And I began by measuring two key physiological traits, the critical thermal minimum and the critical thermal maximum, cold and heat tolerance, respectively. These two traits describe the thermal limits to locomotion, and the way that we measure it is by systematically cooling or heating the lizards and determining the temperature at which they lose the ability to right themselves when flipped on their backs, this being the thermal limit to locomotion. And as you can see, it involves incredibly sophisticated equipment in the form of a heat lamp and a bucket. So it's an incredibly field portable technique. So what I did was, along with an incredible team of undergraduates, scour the island for as many populations of these lizards as we could get our hands on and measure their critical thermal limits. I'm going to show you the results. So on this plot, elevation is on the x-axis, going from sea level to about 2.5 kilometers above sea level, and cold tolerance, or CT-min, is on the y-axis. Each point corresponds to a population mean, and the color tells you the species from which the data were gathered. A lower value on this plot indicates a more cold-tolerant population. That is, if lizards were able to tolerate cooler temperatures before losing the ability to right themselves. And what we discovered was an incredibly strong clinal relationship between elevation and cold tolerance, such that those high elevation lizards were up to 7 degrees Celsius more cold tolerant than their low elevation counterparts. So far, this makes sense. It gets cooler with elevation. Let's do the same exercise, but now with CT max on the y-axis. Again, these are the same populations as before, but now we're looking at heat tolerance. A higher value of CT max indicates a more heat tolerant population. And here's what we found. About half as much within population variation for CT max as for CT min, and absolutely no relationship between elevation and heat tolerance. Whether near sea level or 2.5 kilometers above sea level, heat tolerance hovers somewhere between 38 and 40 degrees Celsius. These results were slightly counterintuitive to us, so we had decided to take a closer look at the thermal environments that these organisms occupy to see if this could help us contextualize our results a bit better. So what we did was visit 4 sites ranging from 45 meters to 2,450 meters to get a deeper perspective of the temperatures available to lizards in those habitats. We deployed a large number of thermal sensors in each of these habitats to record temperatures during both nighttime and daytime. I'm going to start with the nighttime temperatures first. On this plot, time is on the x-axis, going from sundown to sun up. And here are the temperatures that we observed. Very little thermal variation within any given site, about 5 to 10 degrees Celsius variation, and very little, if any, thermal overlap across site. So the temperatures experienced at night at high elevation have almost nothing to do with the temperatures experienced at night at low elevation, and vice versa. Correspondingly, you can sort of visualize the nighttime environment in Hispaniola as something like this, thermally structured into different zones that get progressively cooler with elevation. Correspondingly, the really tight clinal relationship we observed between CT min and elevation makes intuitive sense. Let's do the same exercise, but now with daytime temperatures. So on this plot, we're now going from sun up to sun down, and these are the same 4 sites. Here are the temperatures we recorded. A lot more thermal variation within sites. In fact, about 30 to 40 degrees Celsius variation at any given site. This is a lot of thermal heterogeneity for a lizard to contend with. And importantly, much if not most of that thermal variation was shared across sites. Maybe it doesn't get quite so cool at low elevation or quite so warm at high elevation, but overall you can see that there's a thermal band of shared thermal conditions across sites. Correspondingly, you can sort of visualize the daytime environment on Hispaniola to look something more like this, reflecting a lot more within site thermal heterogeneity and a lot of shared heterogeneity across sites. Now, if CT max varied in a way that paralleled the pattern we observed for CT min, we might have observed more within population variation and some, maybe a shallower, but some clinal relationship between CT max and elevation. But in fact, the data look nothing like that. The data looks something more like this: very little variation and no relationship whatsoever with elevation. And it turns out that there was one key piece of this puzzle that was missing. And in order to get there, Bernardo or anyone else in the audience, would you care to unmute yourself and share any observation, something unusual that you see this Enolus cybotes doing? There's no wrong answers. I've heard them all. Please feel free.
Is it raising its feet?
Yes, that's a great observation. In fact, if you observe, it's elevating its hind foot off the leaf. And if you peer in closely, you can also see that it's curling its foretoes off the leaf. It's using behavior to modify its interaction with the incredibly hot surface of this palm leaf. And the piece of this puzzle that was missing was thermal regulation. But before I get there, it actually is worthwhile taking a step back to explain what I mean by thermal regulation. Even though this concept may seem intuitive or perhaps even obvious to you and me, it's only been formalized as a concept for less than a century. To put this into context, we as a community discovered the structure of DNA less than a decade after the concept of thermal regulation was formalized. So let's take a step back and explain what we mean by it. Let's take a bird's eye view of a thermal habitat, shown here with this forest. Maybe deep in the woods, it's too cold for lizards to thermoregulate. Maybe out in the open, they would actually fry. Maybe it's the flanking vegetation along the forest periphery that's actually optimally warm for lizards. Let's go ahead and color that a very bright orange. And let's envision that a lizard were to simply shuttle around the entire thermal habitat. Such a thermal conformer would indeed experience the highs, the lows, and the in-betweens of the habitat, in effect, all of the thermal variation available to it. But if instead a lizard were simply to preferentially shuttle between the thermally optimal patches of its habitat, it would in practice only experience a relatively narrow subset of the available thermal environment. And in so doing, transform its thermal environment to one that looks something more like this. And that is in fact what these cybotoid anoles are doing. Here are field active body temperatures recorded from the same populations from which we gathered heat and cold tolerance. What you can see is that across 2.5 kilometers of elevation, body temperature only drops on average by about 1.5 degrees. To put this into context, if these same lizards had been behaviorally passive with respect to the thermal environment, it should have dropped by more like 10 degrees Celsius. So in effect, what these lizards are doing is transforming an incredibly thermally heterogeneous environment through their behavior into one that's much more thermally homogeneous, for example, shown on the right. But we can take this one step further. Back in the 1940s, when these ideas about thermal regulation were originally being articulated, one of the major architects of that idea, Charles Bogert, shown here on the right, revisited this notion in a paper in 1949 that published in one of the earliest issues of Evolution, and in my opinion, is one of the most prescient studies of its time. In it, he said, wait a minute, by buffering themselves from environmental variation, organisms aren't just limiting the temperatures they experience in the habitat. In so doing, they're effectively sculpting natural selection. They're shielding themselves from natural selection. When extrapolated over long time periods, such behavioral buffering has the potential to slow evolution and preclude physiological evolution. We now know this idea as behavioral inertia, but you may also know it as the Bogert effect in honor of Charles's pioneering efforts. And in fact, that's exactly what's occurring in these cybotoid anoles. Cold tolerance, a feature that cannot be behaviorally buffered against the local thermal environment, evolves not only more rapidly than the features of the upper end of the thermal performance curve, it can evolve up to an order of magnitude faster. Something that stuck with me, however, through these studies is why are these montane lizards going through the trouble? Why behaviorally adjust to cold environments when you can just adjust your physiology to the prevailing conditions? In other words, why aren't these high elevation anoles more physiologically cool adapted? One possibility is that heat tolerance, for example, might not evolve for reasons completely unrelated to behavior. It may be inert for myriad other reasons. So what I did was gather every datum I could find from squamates on heat tolerance, or CT max. And when I did, I discovered more than 25 degrees Celsius variation in this feature. When we zoom in on anoles, we discover more than 20 degrees Celsius variation. So it's not the case that CT max, for example, can't evolve. It certainly does. Instead, what I suspect is happening is that these high elevation anoles are capitalizing on the physiological benefits of a higher core temperature. All else being equal, when ectotherms perform a task, they can often do so at a higher maximal performance up until a certain limit, if they do so at a higher body temperature. Okay, so up until now, I've been really focusing on the role of thermoregulation as a potential brake on evolution. For example, by limiting exposure to selection, by reducing environmental heterogeneity, and effectively pumping the brakes on physiological evolution. But this is only one perspective on the potential role of behavior in evolution, and if anything, it's not the more classical perspective. If anything, the classical take on the role of behavior in evolution is the exact opposite, which is that behavior should drive evolution. One of the key architects of this idea was Ernst Mayr, shown here. He spent many decades traveling around the tropical regions of the world and trying to uncover the mechanisms underlying the incredible biodiversity of birds that he observed there. And this incredible diversity, in his opinion, was in no small part due to behavior. Behavior allows organisms to exploit new resources within their environments, or even enter new environments altogether, and in so doing, expose themselves to natural selection and accelerate evolution. Now, I don't want to suggest that behavioral inertia and behavioral drive are mutually exclusive concepts. They certainly aren't, but they have been largely studied independently. And one of the things that I was left wondering after those first studies with the cibitoids was, what exactly are those anoles mechanistically doing on the ground to behaviorally buffer their thermal environments so well? And does that involve a shift in resource use that may, as a correlated effect, impact selection in other phenotypic dimensions? In other words, can behavioral drive and behavioral inertia occur simultaneously? So this is what I set out to test. And in order to do that, I visited both high and low elevation sites to much more mechanistically pinpoint what lizards were doing to thermoregulate. In particular, I visited 2 sites at low elevation, one each in the foothills of the Sierra de Bauruco and the Cordillera Central, shown here, and 2 sites at high elevation, one in the Sierra de Bauruco and another in the Cordillera Central. As you can see, these are very different environments, much, much colder. Now, in order to understand whether and how organisms are thermoregulating, you really need 3 pieces of data. First, you need some understanding of the thermal environment available to lizards. In other words, what temperatures could lizards theoretically access in their environments? And the way that we determine this is by deploying a series of copper lizard models. This is literally copper electroformed into the shape of an Enolus Lizards. And inside each of these lizards, we embedded an eye button that measured temperature every 10 minutes at each of these sites over the course of 6 days. We use electroformed copper because it heats up and cools off at a rate comparable to live lizards. That is, it has a similar thermal inertia. And because these are trunk ground anoles, meaning they perch within the bottom 1.5 meters of the canopy, we deployed these models in equal numbers on boulders and on tree trunks because these are the perches that these lizards most commonly access. At the same time that these devices were out in the environment recording temperature, we were focused on the second piece of data that we needed to collect, which is field active temperatures from lizards in those environments. In other words, we need to know what subset of temperatures are being accessed in practice by the lizards. In order to do this, we went out into the habitats and collected live lizards and measured their temperature, this of course being the most fun part of the experiment. And lastly, the last piece of data that we needed was some understanding of the thermal range that lizards were theoretically trying to access in their environments. And in order to do that, we brought a subset of lizards from each of the 4 sites back to the lab and exposed them to a thermal gradient that ranged from 18 degrees Celsius on one end to 40 degrees Celsius on the other. And through these experiments, these lizards were released from any ecological constraints. There were no predators, there were no competitors, there was no food. So this allowed us to determine what thermal range these lizards would theoretically try to access in the wild. And we use these data to calibrate our understanding of whether and by how much lizards thermoregulate in the field. Now, before I walk you through the results, I've created some dummy data so as to prime you to visually know what to expect. So here on the left, I've created a null distribution of operative or environmental temperatures. If the lizard body temperatures overlap in mean and range with those that are available in the environment, we infer that those lizards are thermoconformers. By contrast, if the mean and range of temperatures differs quite substantially from what's available in the environment, we determine instead that those lizards are thermoregulators. And of course, we can put metrics behind this to put numbers behind these qualitative patterns. Okay, I'm going to walk you through the results now, and I'm going to start with low elevation. The western mountains are on the left, and the eastern mountains are on the right. Here are the temperatures we recorded on boulders. Here are the temperatures recorded on tree trunks. Here are the lizard body temperatures. So Bernardo, I'm going to put you on the spot again. Are these thermoconformers or thermoregulators?
They seem kind of conformers to me.
Yeah, that's a very good inference. They are not perfect thermoconformers, but they are mostly thermoconformers. They're quite behaviorally passive with respect to their thermal environments. Now, if we superimpose the preferred range on this, we know exactly why. And the reason is that the low elevation environments are quite optimally warm. It turns out that being near sea level in the Caribbean is not just optimal for humans, it's also optimal for lizards, for these lizards anyway. In other words, the thermal environment overlaps with lizards' preferred range quite often. So in other words, these lizards can access temperatures within their preferred range without having to be quite behaviorally precise. So they don't thermoregulate because they really don't have to. Okay, let's do the same exercise, but now at high elevation. Here are the temperatures measured on boulders. Here are the temperatures measured on tree trunks. Here's our first finding. I promise you this is highly scientific. Trees are colder than boulders. Here are the lizard body temperatures. Thermoregulators or thermoconformers, Bernardo?
Now they seem thermoregulators.
Yeah, incredibly precise thermoregulators, it turns out. When we superimpose the preferred range on this, we can see just how good they are at accessing temperatures that are exceedingly rare in their habitats. If we zoom in on this narrow little sliver of the thermal environment that's actually warm enough for thermoregulation to be effective, this finding that tree trunks are colder than boulders suddenly becomes much more important. And the reason is that about 95% of the temperatures that we measured on tree trunks were below these lizards' preferred thermal range. By contrast, boulders were 5 to 6 times more likely to fall within lizards' preferred range. So put differently, an enterprising lizard could quite effectively thermoregulate at high elevation simply by eschewing tree trunks and opting instead to perch on boulders. And that's exactly what these lizards do. At low elevation, they exhibit the arboreal niche that is characteristic of Enolus lizards, and at high elevation, they've almost completely switched over to become boulder-dwelling or saxicolous. Now, why does this matter? This matters because our entire understanding of the Enolus adaptive radiation is predicated on the incredibly tight connection between structural microhabitat use and corresponding morphological specialization. That's quite literally how we define the ecomorphs. So put differently, when an Enolus lizard alters where in the habitat it perches, it's a reasonable inference that morphological evolution might follow. So our next step in this project was to bring adult lizards back from the wild and measure a few traits that tend to evolve when lizards go boulder-dwelling or saxicolous from an arboreal ancestor. And these distill down to skull and limb dimensions. Now I'm going to walk you through the results first. I'm going to start with head height first. So this describes how relatively tall the skull is. To help visualize the results, I'm showing residual plots here. So on this plot, a relatively larger value will indicate a population of lizards with a relatively taller skull, and a smaller value will indicate lizards that have more dorsoventrally compressed skulls. And what we discovered was that both in the western and the eastern mountains, those high elevation lizards have dorsoventrally compressed skulls. They have relatively flatter skulls. Now, sometimes you don't even need to do the math. I've shown you images here of a low elevation Enolus cybotes and a high elevation Enolus shrivi. And the common name for the low elevation anole is the large-headed anole. So you can even see how different the skull dimensions are for these lizards. Now, one possibility is that these differences in skull height are due to phenotypic plasticity and may not even have a heritable basis whatsoever. So in order to assess this, we brought adult lizards back from the wild from both high and low elevation. We let them lay eggs in the lab, and then we reared those young to adulthood and remeasured the same morphological traits. And when we did so, we discovered that the same differences persisted. Whether caught in the field as adults or raised in the lab under shared conditions, those high elevation lizards have relatively flatter heads. Now, the adaptive value of this shift is quite intuitive. These lizards don't just perch on boulders for warmth. They defend them as territories, they retreat under them when threatened, and they sleep under them at night. They have, in the truest sense, become saxicolous lizards, and dorsal-ventral compression eases access into the crevices that they now occupy. We also detected reductions in hind limb length in both the femur and the tibia, and those differences persisted in the laboratory rear generation. Now, hind limb length is a little bit more complex, but in general, greater limb parity, that is greater similarity in fore and hind limb dimensions, is a feature often associated with greater stability during climbing. And these high elevation anoles effectively spend most of their time scrabbling over cliff faces and perching on very broad boulders. Okay, so what's the broader point here? Well, the broader point that I want to make is not so much that behavioral drive and inertia are both true. We certainly knew that, but rather they may actually be interrelated phenomena. And the reason for this is that any behavioral decision that an organism makes that involves any shift in resource use may simultaneously impinge on multiple selective dimensions at once. So to give an example, when this Enolus cybotes made a behavioral decision to perch on this leaf, it didn't just make a decision about its thermal environment, it actually made a whole slew of behavioral decisions. It made a decision about its radiative environment, its hydric environment. This substrate that it's perching on has a certain height, frictive coefficient, and surface area. So it's also made a biomechanical decision. It's also made a decision about the predators, competitors, and parasites that it may encounter. So put together, these results suggest that any behavioral decision an organism makes may have multiple, and in the case of these anoles, potentially contrasting evolutionary consequences for different aspects of the phenotype. Okay, so at the end of all these studies, I was really struck by the incredibly potent impact of the Bogart effect for behavioral inertia in those cybotoid anoles. This really strong signature of thermal regulation and a corresponding signature on physiological evolution. And something I started to wonder was how generalizable is this phenomenon? Is the Bogart effect, is really strong thermal regulation and slower rates of physiological evolution a cybotoid lizard phenomenon? Is it a Hispaniolan phenomenon, a Caribbean anole phenomenon? Or is it something that might actually more generally characterize the entire Enolus adaptive radiation? So the work I'm about to describe sought to answer just that, and it was led by a former undergraduate in my lab, Giancarlo Salazar, shown here, who's now a graduate student at Washington University. Now, I really want to highlight John's work because what I'm about to describe was effectively his senior thesis, and it was a Herculean task. John collected CT-min and CT-max data from 14 species of Colombian anoles, and he combined those data with every datum he could get his hands on for 3 features in Enolus lizards: field active body temperature, the critical thermal maximum or heat tolerance, and the critical thermal minimum or cold tolerance. And in total, we were able to collect data from more than 100 species of Enolus lizards. So this really was a monumental task. Okay, to start, we wanted to ask a very general question, which was whether the signature of thermal regulation differs between island and mainland anoles. On this plot, mean annual temperature is on the x-axis. This is meant as a relatively crude proxy of the available thermal environment to different species of anole. And on the y-axis is field-active mean body temperature as measured for each of these different species. Blue points will correspond to species from the Latin American mainland, and orange points will correspond to species from islands. And in general, what we discovered was that the signature of thermal regulation was stronger in island species than in their mainland counterparts. Now, behavioral data are inherently noisy. They're subject to the vagaries of weather and many other features. But in general, what you can observe is that mean body temperatures are relatively higher and more stable across thermal environments on the islands. By contrast, and this is perhaps the part that surprised me more, was how cleanly mainland body temperatures seem to track the available thermal environment. Putting these pieces together, this seems to suggest that thermal regulation might be stronger on average in Caribbean island anoles than in their Latin American counterparts. Now, this is exciting because it sets up the possibility that the Bogart effect, this restraining influence of regulatory behaviors on evolution, might correspondingly be stronger on islands than on the mainland. And if so, we had a couple of hypotheses that we could test. First, if the Bogart effect is stronger on islands, we would expect there to be a slower rate of heat tolerance evolution. And given that those island anoles are thermoregulating to a higher core temperature, we might also hypothesize that the evolutionary phenotypic optimum for heat tolerance is also higher on the islands. Now, if it really is thermoregulation that matters here, almost as a sanity check, we would expect there not to be any differences in cold tolerance evolution between island and mainland anoles. So these were the hypotheses we set out to test. And the way that we tested them was by fitting a series of different evolutionary models to the heat and cold tolerance data and to determine whether the pattern and rate of physiological evolution differs between island and mainland species. I'm going to walk you through the models. The simplest of those models was a single rate Brownian motion model in which the phenotypic differences in either heat or cold tolerance among species was simply proportionate to branch length or time, with no differences expected between mainland and island species. A slightly more complex Brownian motion model simply allowed the rate of evolution to vary between island and mainland taxa. The other three models were all adaptive evolution or Ornstein-Uhlenbeck models. The simplest of these models was one in which there was a shared or global phenotypic optimum for either heat or cold tolerance for all species, and a slightly more complex OU model simply allowed the location of the phenotypic optimum, or theta, to vary between island and mainland taxa. In short, there were 5 different models that varied in complexity. We ran 500 simulations for each of these traits and then fitted the model to each of those simulations. And what we discovered was that the evolutionary dynamics definitely differ between heat and cold tolerance. In particular, you don't need any model more sophisticated than a single rate Brownian motion model to describe the differences in CT min, or cold tolerance, among species. There are no differences that we could detect between island and mainland anoles for cold tolerance. By contrast, the evolutionary dynamics of heat tolerance definitely differs between island and mainland anoles, both in the rate of evolution and in the phenotypic optimum. The question is, does it differ in a way that's consistent with the Bogart effect being stronger on islands? Now, as a reminder, if that were to be the case, we would expect a slower rate of evolution on islands, and that those island anoles are evolving to a higher phenotypic optimum. And that's exactly what we observe. Heat tolerance is 3.5 times slower on islands than it is on the mainland, and the phenotypic optimum is about 4 degrees Celsius warmer on islands. Now, if you know anything about Enolus lizards, this finding is somewhat strange because, if anything, Caribbean anoles from the islands are paragons of incredibly rapid evolution. That's part of the story of their adaptive radiation and how these ecomorphs arose. And the mechanism underlying this incredibly prolific and rapid radiation on islands is thought to be ecological opportunity, this release from their ancestral competitors and predators. And yet here I am, here we are, by contrast, describing that when you look at physiology, if anything, those island anoles are actually more slow evolving. So a big question is why? Why are we discovering this island Bogart effect when so many other features appear to be fast evolving on islands? Well, island and mainland anoles differ in many ways, but one of the most salient for this story is movement rates. Now, the first time I ever worked on Enolus lizards was actually with Anthony, who's here, and this was in Costa Rica. And Anthony had us go around the habitats and make video observation of anoles. And I can tell you that the anoles, basically, this photograph might as well be a video, because that's basically what the anoles were doing, which was not moving around very much at all. They spend most of their time standing still and being as cryptic as possible. And I thought naively that maybe anoles just didn't move around a lot. And then I started working on island anoles and discovered, oh no, island lizards actually spend most of their time running around their habitats, dewlapping and head bobbing, and in my opinion, being as visually loud as possible. And this is effectively the hallmark of ecological opportunity. This is that release from predators and competitors that was so important for their ecomorphological evolution. But the ability to move around one's habitat and release from predators and competitors doesn't just matter for exploiting different structural niches, it also matters for thermal regulation. Because the behavioral decision to thermoregulate reflects an inherent trade-off between its benefits, for example, a performance advantage, counterbalanced against its inherent costs, for example, exposure to predators and competitors, which is precisely what these island anoles have been released from. So putting all these pieces together, we come to the somewhat surprising conclusion that ecological opportunity, a feature almost universally associated with faster evolution, can actually slow physiological evolution down by reducing the inherent costs of thermal regulation. Okay, so at this point, these are some of the vignettes that sort of describe what we do in my lab and some of the research that's already happened. But with the few minutes that remain, I just want to highlight some emergent research that's going on in my lab right now. Here's an image of us taken in the before times, so we definitely need to update this image. But we are recruiting new graduate students next year, so I do want to get the word out about that. And I want to highlight two projects that are going on. And even though I can't get into the details here, I will be giving a much longer talk about these projects at SICB 2022 in Phoenix. And the talk will also be broadcasted digitally if you're unable to come in person. Now, I've always, so we still continue to work on reptiles and anoles, but we've also expanded beyond anoles to also work on amphibians, in particular plethodontid or lungless salamanders. Now, I've been fascinated by this radiation for a very long time. I mean, first of all, look at them. They're amazing. For two reasons. First, they're kind of unusual as herp lineages go because they achieve peak biodiversity outside of the tropics, in particular in Eastern North America, which is unusual in and of itself. And secondly, they present somewhat of an inverted case to Enolus lizards because many sublineages within this radiation, for example, the woodland salamanders in the genus Plethodon shown here, are known as a non-adaptive radiation, meaning they're quite species rich in the absence of any pronounced ecomorphological evolution. But research in my lab is discovering that that physiology may be an untapped dimension of diversity in this radiation. We're discovering that species vary in their microclimatic preferences, ranging from some liking it warmer and moister and others liking it cooler and drier. And these seem to be structured along major phylogenetic breaks in the radiation. And currently we've got lots of species of this radiation in the lab, and we're following up these microclimatic studies with examinations of water loss rates, thermal preferences, heat and cold tolerance, and metabolic rates to see whether and by how much physiological diversification may have contributed to this incredibly rich radiation. We're still working on Enolus lizards, of course, and one of the things that I think you may have gleaned from this talk and from many others is that this is a radiation that has predominantly been studied with respect to structural microhabitat. In fact, the plot on the left tells you a lot about what we know about Enolus lizards. But it turns out or rather, one of the things that's emerging especially over the past couple of years, is just how important diversification into different thermal niches has been for the diversity of this radiation. In particular, anoles have extensively diversified into different thermal macrohabitats, for example, across elevation, as well as into different thermal microhabitats along the sunshade axis. And one of the things we're doing is following up lots of studies in Puerto Rico with other studies across other islands of the Caribbean. For example, my graduate student, Brooke Bodensteiner, has been studying physiological evolution on the island of Hispaniola. And as a teaser, what we're discovering is that a lot of the determinism that characterizes ecomorphological evolution in the Caribbean tends to fall apart when you consider physiological evolution across islands. So the physiological dimension of this radiation appears much less deterministic, and we're sort of trying to unpack why that is. So please do come to those talks if you can, and if not, I hope to have much more to share about these projects in the next couple of years. With that, I just want to thank you for your time and for being here, and hopefully take any questions that you might have.
Very well. Je suis sûr qu'on a beaucoup de questions. Alors, je vous laisse l'opportunité de faire votre question. Okay, I'll start then. While the other folks have time to think.
I think someone actually raised their hand, and I think they may have put it back down. Go ahead, Bernardo.
Okay. So this was amazing. Lots of food for thought. So one of the things I was thinking, I mean, you've provided a lot of interesting hypotheses and tests of hypotheses about this difference in the strength of the Bogart effect in islands. And I was wondering if a complementary hypothesis could be because, you know, the islands often have a sort of like more extremes or more specific environments than the continent, and colonization in these islands is often more recent, right? Especially if you look at the Caribbean, these things probably got there by dispersal, and then probably if you were colonized recently a place and you have a different environment to contend with, the sort of like behavior might be your first line of response, and whether that might also be a thing. You know, like in the continents, you'd have a situation in which the species that are there have been adapting slowly their physiologies to those environments for, you know, several million years. But then when you have an environment has been more recently colonized, then behavior is kind of like your first response to that.
Those are both really good observations, and I have a few thoughts to share about them. So I'm going to start with so yes, you were basically hinting at the behavior first hypothesis, really well laid out, for example, in the 2003 paper by Simone Blomberg et al. And in a general sense, that's true. But I should mention that the mainland Latin America has been back colonized by island radiations, meaning that many, in fact, a very rich radiation of mainland anoles are younger species or younger lineages than island radiation. So it's not the case that island lizards as a whole are actually younger. In many cases, they're older. The other point that you raised, however, I think is the really important one, and the one that we weren't able to address in what I would consider a satisfactory manner, which is whether the thermal habitats differ on a much more micro scale level. So using macro scale thermal data, we were able to, we sort of compared macroclimatic patterns between mainland and island lineages, and at coarse environmental scales, we really didn't detect major differences. But other studies have detected differences. For example, there's a great study by Julian Velasco in 2016, and he found faster climatic niche evolution in the mainland, perhaps reflecting a more limited ability to thermoregulate and, you know, other pressures for physiological evolution. But something that I think is generally true, more often true on the mainland than on islands, is that many more mainland species find themselves in generally more close canopied, thermally homogeneous habitats like the interiors of rainforests. So even if you remove the predators and competitors from the equation, and those certainly do matter, those environments are inherently costlier for thermal regulation. So all else being equal, if you still like scrub the predators away, I would still anticipate a generally faster rate of physiological evolution in the mainland. Maybe the signature would be a bit weaker. But it doesn't mean that closed canopied habitats don't exist in the islands. They certainly do. And it turns out in those cases, that's where you find the more thermal conforming types of island anoles. So it's not all about predation pressure. I think those two kind of combine to sort of dictate emergent patterns of thermal regulation, and the thermal environment at incredibly micro scales, like the sun shade axis, a much harder dimension to get for hundreds of species, probably differs in a really important way. So that's a really astute observation.
Merci. On a plus de questions. Violaine?
Yes, can you hear me?
Yes.
Sorry, Bernardo, I cut your microphone because we were listening to the hammer. It was a bit disturbing.
Yeah, no, I understand. There's work being done here in the facade of the building, so yeah, I'm more disturbed than you even probably, but.
But for Martha. So you argue that behavior can be a break into a physiological evolution or thermoregulation evolution in the lizards that live up high in the mountain on the island. But in the same time, you say that you observe this morphological evolution where they are becoming flatter and smaller, if I understood correctly. So somehow this behavior that they restrict their habitat use into the rocky environment does drive morphological evolution. So in terms, if you consider several traits, it's kind of a philosophical question, but I would not necessarily conclude that behavior in that case is slowing down the rate of evolution. It depends on the trait, right?
Yes, it depends on the traits. And we didn't explicitly compare the rates of physiological and morphological evolution. We could. These would be tip-specific rates. I think it's more how do the evolutionary signatures on different phenotypic dimensions vary based on a shared behavioral shift.
Okay.
I'm not saying that morphological evolution is faster than physiological. We would have to sort of put them on the same axis. You know, you can't compare them just as is, right? You have to make them apples to apples and then compare the rate of evolution. So we could do that. That's a great idea, actually, and make it an apples to apples comparison and ask whether one is faster than the other. And in a general sense, actually, we do have a study in review comparing that for all Hispaniolan anoles, and we did put them on the same axis, and you get a mosaic pattern of evolution where so we looked at heat tolerance, body size, and hind limb length, and cold tolerance for 29 species of Hispaniolan anoles. And in that study, we discovered that the slowest evolving trait was heat tolerance, then body size and limb dimensions, and then the fastest evolving trait was cold tolerance. So the morphological rates in that study were actually nestled between the physiological rates.
Okay.
Yeah.
Thank you. It was an amazing talk anyway.
Oh, thank you. That's very kind. It does bring up a point, though. So if I may, while we wait for another question, this raises an observation about, well, you just looked at physiology and morphology, and that's really true. There's so many other features that vary between these anoles. So for example, they perch on tree trunks at low elevation, right? So they're tan colored or brownish. And at high elevation, they're much darker, not quite black, but like very dark colored. That impacts thermal regulation because it impacts the rates of heating up and cooling off. But it also matters for substrate matching because they perch on these different colored boulders, and they're in these really open environments with lots of aerial predators. And one of the other things that differs is flight initiation distance. So if you try to approach low elevation lizards, you can get within like a few inches of them before they try to flee. And when they try to flee, they basically very lazily like run around to the other side of a tree trunk. At high elevation, you can barely get within 2 to 3 meters of these lizards, and they're already fleeing, and they flee a much greater distance. So I've sort of made it binary, but there's so many features that are probably varying between these anoles.
Okay, and also relative to the predation pressure, which is also something very important apparently. If you compare the island versus the mainland habitat or environment. Yeah. Okay, thanks a lot.
Thank you.
Got a question? No, that's fine for us. Thank you.
OK, je pense qu'on a le temps encore pour une ou deux questions, si on y a. Sinon, je profite. OK, on a Étienne.
Hi, Martha and everybody. Thank you very much for this really great talks. So I just wanted to make a small comment. I need to go to teaching just after, but just telling you that I'm listening from the Lesser Antilles right now. So I'm in Guadeloupe. We are Anolis just around, so not right on the campus, but not so far. And we are also working on Hispaniola with colleagues, not on Anolis for the moment, but mostly on fish and birds. That was really great. And I think we could continue discussing a bit later on, or maybe there, on the differences between this radiation in the Greater Antilles and the Lesser Antilles, where we have like only one species or complex species in each island. So in French West Indies, we have Guadeloupe and Martinique with two different species or complex. And I think that could be really interesting. So they are really moving around. So that's maybe some behavior that will be a choice of moving of microhabitats. So there's less differences in microhabitat, less predators also. So yeah, anyway, that's great. I don't think that there's thermotolerance measured in the Lesser Antilles. Anthony did some biomechanics and study, but for physiological, I'm not so sure. In fact. So yeah, so thank you very much. And if you have some hints about that, I'll be happy to understand, to listen.
This is so exciting to hear. So first, the only data that I'm aware of is that actually the Guadalupean anole marmoratus is one of the few thermal conforming species. There's a study in 1976 by Preston Webster and Ray Huey, and they found that they're actually kind of thermal conformers, but they didn't go across the entire archipelago. They sort of worked on Alliaceus, which is a much more close canopied subspecies. The second observation I want to share is that I actually did get to go to Guadeloupe over a decade ago, and I worked on ecological speciation, or lack thereof, in the Speciosus inornatus ecotone on Grand Terre. And I'm guessing that you are on Basse Terre, and therefore you are in the territory of Marmoratus marmoratus?
We are on Grand Terre.
Oh.
So, but just in the middle, so Pointe-à-Pitre, but...
So Speciosus, the blue-headed one, or the brown one? Speciosus.
Mostly the brown.
Oh, inornatus, also beautiful. Yeah, that was the first species I ever worked on was Enolus marmoratus. So this is very exciting to hear. It got me into anoles.
So you're welcome if you want to come back and plan this. So yeah, we have people from ISYEB and from Borea in Guadeloupe. So we are connected to the museum in Paris. And yeah, that would be great. So just the time shift was perfect with this 1 hour less. So it's early morning for us. So we'll start the day, or it's already started. But yeah.
Thank you so much for coming.
Thanks a lot. I need to leave you, but yeah, looking for further discussion. Thanks a lot. Bye-bye.
Small world.
Super nice, Etienne. Okay, so, I think if we don't have any other questions, we can all thank Dr. Munoz profusely for her talk, which I think everybody found very interesting. And I think we'll see you at the next seminars.
Thank you so much.
All right, thank you. Yeah.
Thank you.